Hello, everyone, and welcome back to Creation Myths. Let me take this screen down. We don't need that right now. We'll get to that in a few minutes. Hi, everybody. Welcome back to Creation Myths. We are doing a live show tonight, and I've been looking forward to this one for a while because we're going to do a live demonstration tonight that shows why one of the most common creationist universal common ancestry, and we're going to show why that response is invalid. That's what we're going to be doing here tonight. So I see we've got a great crowd in the chat. Hi, everybody. It's great to see you all here. So here is the setup for tonight. Whenever anybody asks what the single best piece of evidence for universal common ancestry and specifically universal common ancestry of cellular life is, my answer to this is the nested hierarchical pattern of genomic similarity. Right? You've got humans and chimps, and then gorillas and orangutans and apes, primates, mammals, and so on, all the way up through, the universal common ancestor for all cellular life. You can construct nested hierarchies, whether you're using highly constrained gene sequences or less constrained or unconstrained, non-functional, non-constrained regions. Function is actually doesn't matter. It's just the level of constraints in the nucleotides matters. So you can do this for unconstrained regions. You can do it for constrained regions. You get the same pattern of a nested hierarchy of similarity, right? And the fact that that pattern exists is the single strongest piece of evidence for universal common ancestry. Now, whenever you bring this up to creationists, they say one thing every time. The response is always the same. And if you're familiar with creationism, you know what that response is. That response is, well, it could have been created that way. A designer is going to reuse parts in its designs, and if things are similar morphologically, functionally, biochemically, whatever, then the underlying genomics are therefore the gene sequences are going to be the same because a designer is going to reuse parts and they're going to be slightly modified to get slightly different organisms, right? So essentially, the answer boils down to no, that pattern is explainable via common design, not common descent. That's the response you always get. So we're going to test that hypothesis tonight live right now. We're going to do a simple demonstration that anybody can do to show why that response is wrong in terms of a rebuttal to the argument of nested hierarchies, not just being very strong evidence for universal common ancestry, but also directly refuting common design. Okay, so I want everybody to follow along with this if you want to. So down below in the description, there's a link to a random DNA sequence generator. You're going to need that if you want to follow along. And I encourage everybody to, if you're watching this live, you can put it in chat while we're doing it. You can put your results in the chat. If you're watching this afterwards, you can do it yourself and compare your results to what I get and what people in the chat get. So click on that link down below before we go any further, and also have like somewhere to write your results. You're going to be writing sequences and counting. So you could use Word, Excel, paper, and pencil. Doesn't matter. Just have somewhere where you could, tally up your count to see what the results actually are. Because I don't want people watching this to take my word for it. We're going to do it on screen, and I'm going to show you additional replicates that I did myself. But I don't want you to take this on trust from me. I want you to test it yourself and see that you're going to get the same results. Okay? So what we're going to do here is we're going to test the common design and common ancestry hypotheses. We're going to do this using 3 lineages. And basically the idea here is I'm going to label these lineages as human, chimp, gorilla. Some creationists will say that chimps and gorillas are the same kind. Some will say they're a different kind, but I'm using them out of convenience because it's a relationship that we're all going to understand. Okay, so we all know that the existing pattern of diversity is that we have humans and chimps are more similar to each other, and then gorillas are similar but dissimilar relative to the other two. right? So the way you would say that is phylogenetically, gorillas are the outgroup when you're comparing humans and chimps and gorillas. Okay, so we know what the existing pattern looks like, both the pattern of similarity across those three lineages, and importantly, the amount of diversity within each lineage, right? Because what we are testing is how each of these hypotheses, common design and common descent, how well are they able to generate the pattern plus the diversity. Okay, so we're gonna set up a little simulation here, and this is where I'm gonna put up my screen, and we're gonna go through this step by step. And this is where I encourage everybody to follow along with me, because I'm only gonna do it once, and then I'm gonna show you additional trials that I did and show you those results. So I'm gonna do it once, step by step, and I'll show you the results of 6 trials I did, you know, off screen. And then everybody else, you can generate your own data. Check my work. You'll see you're going to get the same results. So let's get into it, folks. This is super cool. So here's what we're going to do. We are going to first generate our sequences right here, which we have not yet generated for the common design model. Now, the way we're going to do this is we are going to be as generous as we possibly can to the creationist position here. The creationist position is that the existing nested hierarchical pattern exists because it was created with a certain pattern of similarity. Well, you can't get any closer to that nested hierarchy than being created with a perfect nested hierarchical pattern of similarity. So for our 3 lineages, which we're going to abbreviate H, C, and G for human, chimp, and gorilla, here is our starting point. And by the way, chat, this is going to be an interactive show. So I'm going to be asking things, asking if you're following me, if that made sense, if I need to clarify. So if you're here hanging out, answer me so I know that we're all on the same page as we go through. So take a look at these 3 sequences right here. The key thing is we have a gorilla sequence. Now, it doesn't matter what these bases are. The point is the pattern of similarity across those 3. So you'll notice gorilla is just all A's, and then chimp is going to be 5 A's and 10 G's, and then human is 5 A's, 5 G's, and 5 T's. And the point here is that this is a perfectly nested hierarchy, right? We have 5 bases, the first 5, that are identical across the 3. Then the next 5 are shared in human and chimp, but are different in gorilla. And then the next 3 are unique in each of the 3 lineages. So we have a perfectly nested hierarchy here. And again, we don't necessarily know if there was common design or if there was separate creation, what that initial state would have looked like. Would they have been created with a perfectly nested hierarchy like this? Would it have been completely random, uncorrelated? Would they be identical? We can't say, right? But what we can say is that in order to generate the existing pattern of similarity, the nested hierarchy that we see, the way to be as generous as possible to the common design hypothesis is to start with that exact pattern. Is everyone following why I did it that way? That we're trying to steelman this position. We're going to give them every benefit of the doubt right off the bat. We're going to start with that pattern. Now, the problem here is that you also need to generate diversity within each sequence or within each lineages. And when I say the problem, I mean the problem for the common design hypothesis. Because at creation, whether you're young Earth, old Earth, whatever, you only have so much variation present in the initial created individuals, right? And this is especially true for the young Earth model where they get off the ark. And if you're talking about apes, you're talking about 2 of each ape kind getting off the ark. So you have at most for each region or each locus or each gene in the genome, you have 4 alleles at most. But there are genes in the human genome, for example, that have over 1,000 alleles. There's 3 main alleles for the ABO blood group gene, right? A, B, and O. But there's a ton of SNPs, single nucleotide polymorphisms, and there's actually over 1,000 different variants of that gene. So in the short time from get off the ark to present day, you have to generate all that variation. And I've actually done the math on this. you can't get that variation due to recombination. Recombination is just too slow to do it. And it's not like you can get half of it via recombination. Recombination is too slow by several orders of magnitude. So the vast majority of the existing variation that is present in each lineage has to appear via mutation. And if we're talking in the young Earth model, which is the main thing we're dealing with, it has to appear via mutation post-flood in that very small population size lineage. Now, this is important because mutations are probabilistic, right? You don't know what mutations are going to happen. They just happen. So the way we're going to model this is we're going to preserve some of that initial nested hierarchical pattern, but we also are going to generate a bunch of diversity. So if you look at this second line here, I've smushed one column from each set of 5 here into the first 3 columns. So you've got AAA, AGG, and AGT. So you preserve that nesting pattern in the first 3, but then N in a DNA sequence just means any base. So we are going to generate new sequences there representing mutations building up post-flood in each of these lineages. So the way we're going to do that is we hop over to right here, our random DNA sequence generator. We're going to set our length to 12. And for the GC content, we're going to leave it as 0.5. That just means that any of the 4 bases has an equal probability. Some genomes are GC biased. You see this especially in prokaryotes. You could have a GC bias as high as 70%. You could also have an AT bias. But mammals tend to be pretty close to 50-50, I think. And we're just going to make it every base is equally likely just to simplify things, even though that's not actually the case. Certain mutations are more likely than others, but we're not going to worry about it. So now we're going to generate 3 of these sequences. So here is going to be our human randomly generated sequence. And if you're playing along, generate your own and copy it somewhere so you have it. And we're going to replace these 12 ends with the sequence we just generated. Then we're going to do it again. Let's get 12 new ones. Here we go. And we'll replace the chimp ends with our new sequence there. And we are going to generate a new one for the gorillas, and we are going to put it right there. Now, remember, what we are doing here is modeling the accumulation of mutations if we're doing a young Earth model post-flood, right? There's a ton of variation within each lineage that couldn't have existed at the exit of the ark because you only have 2 individuals, and since genomes are diploid, you only have 4 alleles or 4 variants for each locus in your genome, maximum. And we know that recombination is too slow. Even if you have a lot of differences in each of those variants, recombination is too slow to mix and match to get all the existing variants that we see in the modern populations. So we need to have mutations accumulating those variations. And since we're dealing with separate kinds, those mutations are going to accumulate independently in each of our 3 lineages. So that's what we just modeled. Okay, so we have our resulting sequences right here. And now I'm just going to, we don't even need this. We can get rid of this. We can get rid of that. And now, let me get rid of common ancestry model. We'll put that on the next page. Here we go. Now we're going to tally up the similarities across these sequences. So the first thing we want to do is say how many sites are the same for all 3. Right? So we're going to look down here and find the sites where it's the same base for all of them. So we've got 1 right here, right? We've got 3 A's, 1 in each lineage right there. And I'm looking through and we're looking for sites where they're all the same, and I don't see any more. So that's going to be 1. Next, we want to look for the sites where human and chimp, that's HC, so where human and chimp are the same and gorilla is different. Okay, so we're looking for a site like this, where, see, they both have a G, and then gorilla has an A right there. So that's 1. There's 2 right there, right? And then we have, looks like 3, 4, looks like we have 5 of those. So we put 5 right there. And then we want to do chimp gorilla. So we're looking at these two rows now. We want to see what the level of similarity is. So let's find ones where they're the same, but human is different. And that looks like it's going to be 1 right here. That's the only one I see right there. We got 1. And then we have the last one is human versus gorilla right here. So we want to see where those are the same, but chimp is different. So we've got, let's see, how many of those do we see? I don't see any. I see 0 of those. And now in total, that's 1, 5 makes 6, 1 more makes 7, because we're looking at 15 nucleotides total. That means 8 of those 15 sites are different in each of the 3 lineages. Okay. Now, what we're going to do here is I said we were going to, CG has 2, you think? Did I miss one? Hang on. Let's see. CG has 2. Hang on. Someone said CG has 2. Let me make sure I didn't miss any. Position 5. 1, 2, 3, 4. Oh, that's HG. HG has 1 right there. Good catch. That's 1. HG has 1 right there. A, T. Yep. Good catch, chat. Thank you for the help on that one. And then that's actually going to be 7, right? Now, what we're going to do here, just to show you the variation, because this doesn't take very long, is we're going to do this again. Let's do another 3 and do that comparison again, right? And let's see what pattern we get. We have plenty of time. This is a quick thing to do, so why not get more data, right? So let's generate 3 more. Here we go. 1. We're going to replace all but the first 3. Okay, and then let's generate another one. And we're going to replace all but the first 3. And we'll generate 1 more. And we're going to replace all but the first 3. All right, so let's do that again. And for round 2 here, let's tally them up. So how many HCG sites do we have? We've got the 1 right there. We've got 1 right there. So we've got 2 this time. And that's it. So we've got 2. For HC, let's see. We've got 1 right here. Here, we'll highlight those lineages so you can see them. We've got 1 right there. We've got, oh, I missed one of the 3s right there. So we've got 3 actually here. And then that's it. And then for CG, let's see how many we have. We have 1 right here, 2, 3, 4, 5. We've got 5 for CG. What did I say it was for HC? Did I count those? We got to count HC. We got 1. We have 1. It's 1. Everyone check my numbers. And then for HG, I'll just highlight the H here, but we want to jump down to the G. Let's see, we've got 1 right here. That's the same. Let's see. And it looks like that's it. It's 1. So 1, 5, and 1 make 7. That's 10. So then we've got 5 that are independent, right? So you can see we do a different trial. We get very different numbers because why? It's random. So those sequences that are accumulating in each lineage independently are uncorrelated with each other. Chat, should we do 1 more or should we keep going? Should we want to go down to common ancestry or should we do 1 more just to get 3? What do we think? Everyone, you tell me. Like I said, this is an interactive show. I'm happy to do 1 more if everybody wants to do 1 more. I'm going to do 1 more because why not? And we'll see how the numbers are consistent versus inconsistent across the different trials. And yes, we're counting all, we're not counting the HCG, right? That's just labeling. That's just labeling. But we're starting here is where we're counting, starting with that first one. Okay, so let's generate 3 more groups of 12 here. So we've got 1. We've got 2. And we've got 3 sets of random mutations that have accumulated once they stepped off the arc. So now let's do this again. For all 3, right? We're comparing across all 3 here. We've got 1. We've got 2. Okay, we got 2. For human and chimp, but not gorilla, we have 1. We have 2 right there. And we have 3. We have 1 right there on the end. We have 3. For chimp and gorilla, right? For chimp and gorilla, we have, let's find out. Looks like I've got 1 right. No, we got 1 right here. Yep, we got 1 right here. And that's it. It's just 1. And then for human and gorilla, we have, doesn't look like we have any. I don't think we have any. It's 0. We're saying it's 3 for HCG. I see 1 right here. I see 1 right, the first one, the A's, and then we've got, oh, we've got another A right there. Oh, and a T. You're right. It is 3. Good call. There we go. So it's 3. So it's 3 for that one. So it's 3 and 3, 7, which means we have 8 that are going to be random. There we go. So there's our 3 trials right there. That's what we've got. Okay. So now let's model the common ancestry hypothesis, right? Let's do a similar thing, but now we're going to have common ancestry rather than common design, right? We designed the nested hierarchical pattern here, but in the second approach, we're going to presumably generate it. So let's see what happens. Okay. So the way we're going to do this is we are going to generate these sequences by generating a random sequence of 15. Okay, and we're going to say this is going to be the gorilla sequence. We're going to take 15 bases, and that's going to be our gorilla sequence right there. Now we're going to take those first 5, 1, 2, 3, 4, 5, and we're going to copy those into the chimp and the human because that represents variation that occurred in the common ancestor of all 3 before they diverged. So whatever mutations happen in there, they're all going to share them. Is everyone following why I did it that way? So we're representing with those with that first group of 5, that's mutations that occur in the common ancestor of humans, chimps, and gorillas. They're going to share that variation. That's why we're doing it that way. Now we're going to generate a sequence of 10, and we're going to fill in the chimp sequence with that. We're going to put this right here. And we're going to take 5 of these right here, and we're going to copy them up into the human, because those second 5 right there are going to represent the regions that are common ancestry in the human and the chimp, but separate in the gorilla. So after the first 5, the gorilla diverges. Human and chimp are still together, so they're going to share the next 5. And then the last thing we need to do is we need to generate 5 more just to fill out that human sequence independently from the other 2. So those last groups of 5 in each line represent the variation that has occurred independently in each of those lineages. So now let's tally up what the pattern of similarity looks like here. So for all 3, looking at human, chimp, and gorilla, we have 1, 2, 3, 4, 5, 6, 7 right there. We have 7 that are similar in all 3. Then for human and chimp, but not gorilla, we have 1, 2, 3, 4 right there. For chimp and gorilla, but not human, we have, looks like 0. Nothing, no, no. Yes, but it's the same. No, yes, but it's the same. No, no, no. And that's all the same. So that's 0. And then for human and gorilla, last one, we're looking for the same in the top row and the bottom row, but different in the middle. We've got one of those right there, T and T, and we have the last one, C and C. So that's going to be 2 right there. And then that means there are 2 sites where there is no similarity because it's 7 and 4 makes 11, 12, 13, 14, 15. Okay. Now, just like we did 3 trials up above, let's do 3 trials right here. So let's do that again and see if we get the same pattern. Now, remember, we're predicting a nested hierarchical pattern where gorillas branch off first, and then human chimps diverge. That's what we're expecting to see. So let's see if that happens again. We're going to do 15. We're going to generate that. And what I'm going to do is actually, to make sure I don't mess anything up, I'm going to paste that in, and then I'm going to completely erase the human and chimp ones just to make sure. So while I'm doing this, how does everybody feel about what we're doing here? Does everyone see why we're doing this modeling the way we're doing it in comparison to what we did for common design? With this one, we're not starting with any inherent pattern. We're just letting mutations happen with varying degrees of common ancestry and seeing what pattern that's going to generate. So chat, let me know if we're feeling okay about this. Okay, so we're going to take our 10 bases here and round out the chimp right here. And then we will take these 5 and copy them up into human. And then we need 5 more. And we'll just stick those onto the end. Okay. So now we could do the same thing for trial number 2. So here we have 1, 2, we're doing all of them now. So we're looking for 1, 2, 3, 4, 5, looks like 6, 7, because just coincidentally, they match the gorilla right there. So we've got 7, and we don't have any more. So we've got 7 again. That's funny. And then here we're looking for human chimp right here. We could ignore, we know that the first 7 were all 3. So we've got 1, 2, 3, and that's it. Oh, and I see a bunch of C's right there. So we actually have 3 of these, but we have 8 of those. And then for chimp gorilla, let's see how we did. We have 1 right here. And that's it. We have 1. And then for human and gorilla, we have 1 right here. We have 2. We have 2. There you go. And that's 8, 9, 10, 13, 14. So there's 1 where there's no similarity in any of them. So since we did 3 up above, let's just do 3 here one more time. Okay. And we'll do the same thing again. So let's go back to doing 15 here. Let's generate our sequence. I'm just going to do it real fast. We'll clear out this one, and we'll clear out this one. And now we're going to generate 10. Oh, whoops. Well, we'll generate the 10, but we need to copy our 5 over first. So we copy our 5, put it there. That's the variation that built up pre-divergence. Now we have our chimp stuff right there, and we're going to take these 5 and put it right there. And now we just need our unique human variation, or I should say independent human variation. It's not necessarily going to be unique though. So let's see what's going on here. Let's count them up. 1, 2, 3, 4, 5. 5. It's just the first 5. For human and chimp, we've got 1, 2, 3, 4, 5, 6, 7 for that one. Chimp gorilla, we are looking at 1 right here. And then for human and gorilla, let's see what we get. We are looking for the same in the top and bottom, different in the middle. And we have 1 right here. And we have 1 right here. So we have 2 again. Remarkably consistent for that one. That's funny. Okay, and that's 5 and 7 is 12, 13, 12, 13, 14, 15. Are there no sites where they're all different? That's funny. In all the times I've done this, that's the first time that turned out to be 0, I think. I don't think I had a 0 for that for any of the other trials. Just goes to show you that when you're randomly generating sequences, you don't know what's going to happen. So now what we want to do is compare these. And, we can actually do this in Excel if anybody, we can just make this comparison. So you know what? Let's just go through it. Let's just do it step by step. And then I'll show you the other data and we'll see how what we did right now compares. But you know what? Let's just do it. Let me pull up a blank worksheet in Excel and we're just going to do it right here. Now we don't need the website anymore. So I'm going to put this right here. I'm going to blow this up so everybody can read what we're doing. Okay, we're going to make this nice and big for us. And we're going to say, we'll do HCG. Is that big enough? I'm going to make it bigger. We're going to make this nice and big so everybody can read it. Okay, so we're going to do HCG, HC, CG, and HG, and then none, right? And this is going to be design, and this is going to be ancestry, and we're going to pool these numbers together. So everyone, I'm going to attempt to do arithmetic in my head live on air, which is a mistake, but I'm going to do it anyway just to save us the time of having to fill in the cells and add them up. So everybody help me out here. We have 1, 3, and 3 makes 7 total. We've got 5, 6, 7, 8. That's going to be 9. And then we have right here, that's 7 again. Here we have 2. And then here is 7 and 5 is 12. That's going to be 20. And that should add up to what? That should add up to 45. That should add up to 45. So 29, 36, 37, 30. That adds up to 45. We're good. Okay. Now let's tally these up. So here we've got 15. That makes 20. For human, chimp, gorilla, here we've got 14. We've got 2. We've got 6 and 3. So now we can graph these. We are going to make a graph. So let's do that. We're going to do this. We're going to do this. Here we are. I'm not going to like do the thing where I would make this look nice, but what you can see here is for common design, the overwhelming most common outcome is right here, right, where there's no correlation among those sites. And I'm not going to take the time to do the statistics, but just eyeballing it, I bet there's no significant difference here. And there's probably a significant difference between these two, probably not across these. Now, when you're looking at ancestry in orange, we can see that all 3 and then human chimp exceed the other 3. And with this sample size, would these differences be statistically significant? I don't know. I genuinely don't know. We're not going to sit here and like do statistics on these, but you can see how in the common ancestry model, you generate a clear, consistent pattern where human, chimp, gorilla and human chimp tend to share the most diversity, and the other pairs or uncorrelated is much less. Whereas in the common design model, having uncorrelated sequences is the most common outcome, and having similarity across those lineages is less common. Now, this was with 3 trials. I've previously done this with 6. So I'm going to show you that. I actually have that stuff ready to show you. So we have a data set that's twice as large as this, and you'll see that it's remarkably consistent. So here are those data. Again, it's replicate 1, 2, 3, 4, 5, 6, and pooled. This is for the common design model on top and the common ancestry model on the bottom. Rather than go through like spreadsheets of numbers, I'll just show you the graph. That's nice and easy. Here, let me make it nice and big for us. I'll just show you the graph. And you can see here the exact same pattern where with common design, you tend to get lots of unique variation within each lineage because that variation has to accumulate independently when they like get off the ark or whatever, right? Whereas in the common ancestry model, you see the most similarity across human, chimp, gorilla, and human chimp because those are the lineages that share common ancestry. Now, the cool thing from this is that you could actually derive phylogenetic trees. Now, you could do this with like a computer program. We're not going to do that. We're just going to kind of eyeball it because small sample size, and we're just doing this, you know, for funsies on YouTube. But if you were to take like actual legitimate, you know, sample sizes and sequence lengths, like 1,000 instead of 15, you could do the same thing that we just did and do this for real using, you know, phylogenetic software. But what we can do is we can just say, given this pattern of similarity, what types of phylogenetic trees do we get? And this is what you get, right? So using the common ancestry data, you get a branching pattern that is a nested hierarchy. So you have humans and chimps as each other's sister taxa right here, with gorillas as the outgroup. You have a bunch of variation due to their common ancestry. And then you have a bunch of variation that's shared in the human and chimp common ancestor, but is separate, independent from the gorilla. And then you have a bunch of diversity that's unique to each lineage. When you're doing common design, we're starting with this pattern. Remember, if you took those starting sequences, you would have built this pattern in the common design model. You build the exact same phylogenetic tree, but that ignores the other half of reality, which is the diversity that exists within each of those lineages. You have to generate all the extant diversity. And since recombination is too slow to do that, and I actually did the math on that in my video from a few years ago on created heterozygosity, I actually show you the math that shows why recombination is too slow. You need mutation to generate that existing level of variation within each lineage, which means they're going to be uncorrelated with each other, and you get a phylogenetic tree like this. This outcome where you have a node that splits 3 ways, that indicates that you can't resolve the relationship between those lineages, right? We can't say whether these two or these two are more closely related to each other and the other one is the outgroup, because the pattern of similarity doesn't tell you a consistent story about their evolutionary history, right? So the word for this, when you get an outcome where you have what's called an unresolved node, that's called polytomy. This is what we get when you have a common design model. Common design models result in low resolution. And if you were to do this thing called bootstrapping, which is where you take a subset of your sample and build the tree with just a subset rather than the full data set, you would get all different trees when you do your bootstrapping. And each one of those trees would have very low, what we call branch support, right? It just means a certain tree only appears in a small fraction of the subset. Whereas in this example, if you were to do this like for real and bootstrap this tree, you would get very high branch support for each of these nodes, which is to say those nodes consistently appear in the pattern that we see across all the different sites that we are comparing, right? So this is the demonstration that shows conceptually why the common design rebuttal does not address the strongest evidence for common ancestry being the nested hierarchical pattern, right? So if anybody in chat has any questions, I would love to take them. I see one question right here. As a reminder, why did we start with 3 nucleotides? Oh, hang on. Why do we start with 3 nucleotides in the common design model? And the answer for that is that we have to generate enough variation in the lineages post-flood, post-arc, post-creation, whatever, right? If they're created with a nested hierarchical pattern, great, but they're not going to have a lot of diversity within each lineage. You have to generate that diversity. If they don't share common ancestry, that diversity has to come from uncorrelated probabilistic mutations within each lineage. So we modeled that by starting with the perfect nested hierarchy, but then only preserving that in a little bit and modeling the rest as generating the variation that's unique, that's happening independently in each lineage. Good question. That's there's a reason we did it that way. And you could do you could preserve less of the initial pattern, you could preserve more of the initial pattern. It depends on the part of the genome, right? Certain regions are going to be more tightly constrained, and you would, in theory, preserve more of the pattern there. Other regions are completely unconstrained, and you would completely obliterate the pattern, and it would be gone, right? So by picking 3, I'm kind of picking a middle ground between preserving a chunk of the pattern and preserving none of it. Yeah. And then I just want to pop this one up here. So like the diversity is also nested. Yes, exactly. When some of the diversity that we observe occurred in the common ancestor, right? You're going to see that shared across the 3 lineages. But when certain mutations happen independently in each lineage, we don't expect them to be correlated. And a really good example of this specific thing is with endogenous retroviruses, where we can have an endogenous retrovirus, a viral integration sequence at a specific place in the genome. Not only do we find those in the same place across the human, chimp, and gorilla genomes, but we can actually find the same mutations at the same place in there. So, like, for example, you might have an endogenous retrovirus that's in the same place in the human, chimp, and gorilla genome, but there might be a specific single base substitution that's present in the human and chimp, but absent from the gorilla within that specific ERV sequence, right? So we're talking about the pattern of similarity across those kinds going back to the common ancestor, but you also have to look at the polymorphisms within each lineage and generate that amount of diversity over the course of its evolutionary history. Some of that diversity that you're going to generate will occur in the common ancestors, and some will occur independently. I hope that addressed what that was responding to what was on screen, but also various other things people in chat were saying. So chat, I hope that addressed your questions. So folks, I think that is where we are going to leave it for tonight. We don't have to belabor the point. And again, the point is that if you say in response to the question, what's the single strongest piece of evidence for universal common ancestry in evolution? If the answer you give is the nested hierarchical pattern of similarity across all cellular life, when you do genomic comparisons, the creationist is always going to answer, aha, but that is just evidence of common design, or I could explain that with common design. And what we just demonstrated in a very simple way that obviously makes a whole bunch of, simplifying assumptions, but you can do this for real. Like, there are papers that have done this for real. But what we just showed is that no, common design doesn't explain that existing pattern, because there's no way, starting from a perfectly nested hierarchy, there is no way to get both the pattern of similarity that we see and the level of diversity that we see within each of those lineages. Therefore, common design is unable to explain the pattern. So there's kind of two aspects to why this argument works. One, the pattern that we observe is an explicit prediction of common ancestry. Common ancestry, if true, must necessarily show this pattern, and it does. But also, this pattern that we observe is impossible under a common design framework unless you want to have some supernatural tinkering going on in an ongoing fashion. And most creationists don't want to do that because they recognize that such a position is unfalsifiable, right? If you're saying like miraculous intervention in what mutations happen occurs, all the time, then we're no longer doing science, right? You can't test that at all. So that's where we're going to leave it for tonight, everybody. Thank you all for being here and going through the simple demonstration with me. Again, I want to encourage everybody to do this yourself. You could do it. You saw how fast we were able to do 3 trials for each model. You could do 10 really fast. And you could just generate the data and see what you get. And if you do that, email me. Take a picture of you'll send me an Excel spreadsheet. Take a picture of a graph you make, right? Email me and, maybe we'll do a follow-up and we'll pool everybody's data and see what we get. That would be a really cool kind of follow-up project. All right. Thanks, everybody. Have a great night. Stay safe and healthy, and I will see you next time. Bye, everyone.